Thanks for inviting me. Can you hear me okay? Okay. All right, so there's my mug up there. That's my official mug. I am a clinical assistant professor at Towson University. Let me tell you a little bit about how I got here. So I did my master's at the University of Alabama at Birmingham in tropical mangrove sponges. It's not related to herps at all. But then I went up I-65 to Purdue University and started working on box turtles for my graduate work. And then in 2016, we came to Towson University. So we've been here almost 7 years now. It's hard to believe. So I work on lots of different projects. They're mostly in herps, sometimes the somewhat derived herps with birds. And I'm often interested in projects that use genetic tools or other molecular tools to try to get at natural history questions. For example, this is my graduate project in box turtles, and the map you see over there is in relatively intact hardwood forest in southern Indiana. Box turtles are, even though they're in this family of river tortoises, they're really more of river turtles. They're really more of a tortoise. They live in this hardwood forest, and it's relatively pristine there. And over the course of a few years, I and lots of helpers picked up something like 700 turtles. Each little triangle is a capture there. And the box turtles get about this big, and then they stop growing, and we don't really know much about what they're doing when they're small, because it's hard to put little radios on them. So we don't know what they're doing or how far they can go. And then when they get adults, they're nice and convenient because you can put a little radio transmitter on them, follow them around. So what we know from adults is that they have these small little home ranges as adults, maybe the size of this room, maybe the size of this building for the most part, and they will stay there for decades. They have everything they need in that little spot. So that would suggest that you could have a population of box turtles in a really small, say, forest. But nobody had ever tested that on the genetic level. If you extract some DNA from these guys and sequence them and ask those questions of the DNA, you can learn a lot. For example, out of these 600 turtles, and this is about 24 kilometers, so not a great distance for a turtle that can really get up and move if they want to. Occasionally we see them moving a couple kilometers a year. You can basically do the daytime TV trick, where you sequence them all, and then you calculate a DNA fingerprint, and you see who's related to whom. And you would think that if box turtles, grew up in one little plot of forest and then stayed there the rest of their lives, you would expect a lot of them to be related, right? Parents and siblings and things like that. That is not at all what we found. Out of those 600 some box turtles that we sequenced for this, we found maybe 5 or 6 that looked like parent-offspring pairs. And for a couple of those, we found them like 20 kilometers apart. So we don't know how they got there, but it appears that there may be some sort of dispersal event that may happen when they're really small, like think of like sea turtles going back out and hanging out for a few years or even a decade before they're mature. So that's the kind of thing I like to do with herps and DNA. Here's another project I worked on. This one happened at the Maryland Zoo. There's a sort of unfortunate natural history experiment where a virus called ranavirus came through their collection of turtles. Anybody seen their collection of turtles, box turtles at the Maryland Zoo? There are about 20 of them, and they're in a little pen that's maybe half the size of this room. So it's a pretty high density. Somebody got ranavirus, and it spread to all the turtles. About half of them died, and about half of them did. And since they're all in the same environment and getting the same veterinary care, we wondered, well, is it something genetically encoded that allows some to survive and some that aren't? If so, then everybody who manages a box turtle population would probably like to know that, so they can sequence their turtles and see if they're susceptible to ranavirus infection or not. We did not find any genetic evidence for any difference between the survivors and those that succumbed. So probably some differences in their baseline health are responsible for it, but with turtles, it's really hard to do baseline health assessments. For one thing, they're largely encased by their shells. And they also don't display disease signs in a lot of cases until it's almost too late. So this figure is just showing over time the proportion of turtles that were testing positive for ranavirus. And as you can see, by day 41 or 42, they're all testing positive in this particular population. So in this case, the DNA that we're using is the viral DNA, not the turtle DNA itself. Here is another interesting study. Usually I ask conservation questions, but this one was slightly different. So this is in double-crested cormorants. These are a poster child for getting almost knocked out by DDT, the use of DDT in the middle of the 20th century. After DDT was banned, their populations rebounded with something of a vengeance. So now they are considered a pest species in much of their range, especially in the southeast, especially if you raise fish. These guys eat fish and they can eat a lot of fish. So in places like South Carolina, the Fish and Wildlife Services manages these depredation permits that allows people to basically go out and blaze away at these things. And so Fish and Wildlife Services, even though they're allowing permits to kill something, they can't just issue a permit to everybody who wants one. They need to have an understanding for what's happening. And one thing they need to know is, are there certain populations they need to manage separately, or are there different subspecies they need to manage separately? 100 years ago or so, before the advent of the molecular stuff, the experts thought that there were these two subpopulations. There was sort of a Florida subpopulation down there, and then there were the true cormorants up here to the north. Auritus. So everywhere where there is a plus sign or a triangle, we got a sample from a cormorant, and we extracted the DNA and we fed the DNA into a computer and basically said, where do the populations fall out, without telling the computer where we actually got the samples. So we didn't put in like the GPS coordinates. And what the computer came up with is what we see in that double line and the sort of dashed line. It decided that there were two populations that sort of follow the flyways. So when they migrate, they either come down the Mississippi, that's one of their main flyways, so west of the Appalachians, or they come down along the East Coast, on the east side of the Appalachians. And so it makes sense that that's how the populations are kind of subdivided. But notice that is totally at odds with what we thought were the subpopulations, the Florida ones and the ones up the north. So this was new information that we can give Fish and Wildlife Service and say, instead of managing this way, you should manage this way when you're issuing permits to control these populations. These days, who's heard of the microbiome? Anybody want to hazard a definition for the microbiome? So what the more and more we look into this stuff, the more we learn that pretty much every multicellular organism is not just one single organism, it's a community. Us, for example, we're made up of about 1 trillion cells. We have about 10 trillion cells of microorganisms, bacteria, fungi, archaea, other little things that live on us and in us. So you can think about yourself as a sort of community walking around, not just an individual. That's normal, that's okay. You don't have to go wash your hands right now. A lot of these things that live in us and on us are really helpful. A lot of them live at the sort of border between the environment and our insides. For example, we have a lot of bacteria in our guts that help us metabolize vitamins, for example. And without them, we would be in trouble. And we have bacteria that live in our throat that help keep out the bad strep, for example. They just take up all the space. We know most of that about that in humans, because that's where we tend to spend the money on this kind of research, but it's now sort of trickling down to wildlife. This was a project I worked on in the hellbenders. So hellbenders, there are two subspecies of the hellbender. The hellbender is North America's chunkiest salamander. They can get up to about this big, and they live in like trout streams, highly oxygenated, cold, clear, watered streams. In Maryland, depending on who you ask, we still have a few relic populations way west, like Garrett County. These two subpopulations only coexist in the Ozarks. And what's funny about them is when you go and catch an eastern hellbender, they look fine. And every time you catch an Ozark hellbender, and they have a much smaller geographic range, you find all these lesions on their skin. And sometimes they're missing toes. Sometimes they're even missing their whole hand or their whole feet. And a lot of the movement they do is with their tail, so they're surviving. But we wanted to know what is living on these poor things that makes some of them look awful like this. So in previous experiments, when you wanted to know that, you would go out and catch the animals and take out your sterile Q-tip and you'd swab that lesioned area, and then you'd swab it on a petri dish, just like when you go to the doctor and they swab your throat and then tell you to call back in 5 days because they're waiting for the bacteria to grow up. And that's fine, but almost everything out that you would find out in nature doesn't grow on the petri dish. So a few things will grow up and you can identify them, but you still don't really know what's happening. But so a new approach is to take your sterile Q-tips out there and swab the hellbenders, including the parts that are necrotic or where the tissue seems to be dying. And then you extract all the DNA off that swab and feed it into the sequencer, and then the computer, and it tells you, oh, these are all the bacteria that live in there. You can do this with viruses. You can do this with fungi. And you get these sort of charts like this, and they just tell you the proportions of these different kinds of bacteria that are in there. Sometimes you can use this to identify known pathogens, but in a case like this, we didn't really know what to expect. So we learned a couple of cool things about this. One is that you can just sort of eyeball the differences between these two. So these are the swabs we took from the necrotic tissue on the hellbenders. And these are the swabs we took from their back, which we assumed was healthy skin, appeared to be healthy skin. And there's not much difference. So whatever is causing those wounds on the digits or on the hands is probably there's not some particular pathogen that's responsible for that. We don't quite know what is, but we do know from some other genetic work that the Ozark hellbenders are a lot more inbred. So it might be something to do with their immune system that is responding more poorly to the bacteria that live on them than, say, the Eastern hellbenders. So these are swabs from the backs of Eastern hellbenders. These are swabs from the back of Ozarks. Nowhere do they actually live in the same river together, but they live in the same kind of karst areas in the Ozarks where the water chemistry is pretty similar. So this is quite a striking difference. Okay, and then the latest project I'm working on is another microbiome project. This one is back in box turtles, where I have samples from 5 different sites in Illinois and 1 site in Kentucky. And these are figures that can give you an idea for the different things that we're finding on. This is research in progress. And this is the first work to our knowledge in box turtles in the microbiome. So we don't even, if you, in human medicine, we kind of know what is normal and what is not normal for a microbiome. We don't even know what's normal for box turtles, for almost all species in the world. But we can use all these tools that we're getting from human medicine and apply it to wildlife conservation. So perhaps you can go out into a population and swab them and sequence them and try and maybe get an idea for if that's a healthy population or not, just based on their microbiome. Okay, but I'm really here today to talk about the northern map turtle in Maryland, and talk about some challenges and some successes in this. So the first caveat I should tell you is that a lot of this work has been done by Rich Siegel. He is sort of my predecessor there at Towson University. He's retired now, and so he's handing off a lot of this stuff to me. He has done all this beautiful work on the ecology of this population of map turtles, but he doesn't do molecular work. So I'm starting to bring in some projects where we're using some of the turtle DNA and also some of the microbiome DNA, as you might have guessed by now. So first, let me introduce you to northern map turtles, if you're not familiar with them already. They're so called because they have this sort of mappy patterns on them. They are a member of Amididae, the family Amididae, along with box turtles, painted turtles, bog turtles, spotted wood turtles, etc. They can get pretty hefty, up to about 3 pounds, about 11 inches long. They live mostly in large rivers, but they're also found in the Great Lakes and ponds, etc. Pretty much anywhere that has suitable substrate for their prey, which tend to be mollusks and arthropods. They have a relatively large range. In much of their range, they are not, in much of their range, they're considered least concern. But over here in the Susquehanna, it's sort of a separate distribution. It's separated from the main body of the range. And they are listed as a species of special concern in Maryland because there's just a tiny little bit of the Susquehanna in Maryland. So from Maryland's point of view, we have a pretty small population that is sort of bumped up in its protection by listing them as state endangered so that they enjoy more protection, more study, more funding. I also learned recently that there is an introduced population in the Czech Republic, probably from pets. They have strong jaws and mouth parts that are really useful for crushing hard things, especially the females, which are much larger. I'll talk about that in a minute. They have a mostly carnivorous diet. They like to eat things like mollusks and arthropods. Up at the top there, top left, those are zebra mussels, and so called for their sort of zebra shapes. In the same genus, the larger one that they're on is another mussel called a quagga. They will also eat those. Bottom left is the Asian clam. It's another invasive. And then they often eat the caddisflies, that you can see over there too, and crayfish. At least this is what happens if you read the book about what map turtles are supposed to eat. More on that in a bit. Map turtles are what we call sexually dimorphic, meaning they come in two different, they're different based on sex in some way. We see this across the animal kingdom. And in a lot of turtles, we see it. Sometimes the females are larger, sometimes the males are larger. In this family, in particular in map turtles, there are about 13 species in this genus. The females are huge compared to the males. Perhaps because the larger females can lay more eggs, and also perhaps because the males don't tend to do any battle. Like tortoises, land tortoises and things, they will often battle. And so there's selection for them to be larger. But with the male map turtles, it doesn't appear that they fight for mates or fight for territory, so they can be small, and there's not much selection for them to be bigger because it just costs more to be bigger, right? You have to eat more. Females can be twice as long and up to 10 times as massive as the males. Females have a wider head, even relative to their larger size, and so they can crush larger and harder food items. Here are some data from the Susquehanna population from diet analysis. The diet analysis is a polite way of saying that we caught some turtles and put them in tubs, and when they pooped, we went through their poop and tried to see what we could see under a scope, under a microscope. So I didn't do this, somebody got really good at seeing a little bit of a shell and figuring out, oh, this was a zebra mussel, or this was a Asian clam, or something like this. So when you have all this different kind of data, you can squish them into these kinds of figures so that they're in 2 dimensions. The males in this case are in the empty circles, and the females are in the filled circles. And what's important here is that we can see that they cluster. That just tells us that they have different diets. The males tend to eat along the small gastropod. Corbicula is that little Asian clam that's invasive. The females eat some of that too, but they're staying away. They're more into the small gastropods, not into the corbicula. What's missing from this plot, this came out in 2013. What's missing from this plot is the zebra mussels, because at this time they were just barely being detected in the Susquehanna. So none of them were found in the poop from these 40 or so turtles from this study. Not that much longer, so this paper is only 8 years later. You can see that the diet of everybody has changed a lot. So if you look over to the far right, you see adult females. They're mostly eating zebra mussels. And they're also eating some what are called mystery snails, another invasive species, probably from the aquarium trade. And then Phylactolomata, that's a little bryozoan. was not in, nobody knew that the turtles were eating a lot of those. So for the bryozoan and for the mystery snails, males and females are both eating those, but the females in particular have really switched their diet to zebra mussels. Zebra mussels are invasive, so there's a little bit of hope here that maybe so that at least something can eat them, maybe control them, although we haven't seen a huge increase in, say, the map turtle population in the Susquehanna since the introduction of the zebra mussels. Okay, another natural history trait important for our discussion today is reproduction. This just gives us an idea over time about the phenology of map turtles. Phenology is just like when things bloom or when things come out of hibernation. For the turtles, what we're seeing here is number of records of them being observed. So most of the year, much of the year, you're not going to see them because it's too cold and they're chilled out at the bottom of the river. Or like late summer, the water is warm enough, they don't need to come up and bask. We just don't see them. But around May, starting in April or so, they'll start to come up and bask. The females in particular really need to bask because they're doing what's called staging. That is, they're getting warm by being out in the sun, and that speeds up their metabolism, and so they can do things like make yolk for all the eggs that they're harboring. And then about late May, they really start to come up on, the females only start to come up onto the land to lay their nests. And they're often gregarious baskers, which is just a fancy way of saying when one of them finds a good basking spot, a lot of them will come out and hang out too, sometimes even on top of each other. And here on the right, we have a picture of a female who was either on her way to her nest or had just laid her nest when she was caught here on a beach for a picture. What about the nests? Uh-oh. Oh, there we go. So not all of these last half of May through early July records are of females nesting. Many of them are also the hatchlings that are coming out and also being seen. So the way the biology works for map turtles in most places is that the June is really their favorite month for the females to climb out of the water, and they lay their nests, and then probably late summer, early fall, the hatchlings hatch out. And then most places, especially here in Maryland, they chill out. They stay down there in the ground until the next spring. So at the same time we're seeing females in May and June crawling up the beach to go lay their nests, you're also seeing hatchlings coming out and trying to make it to the water. So there are hatchlings in the ground pretty much all year, which has strong management implications. It means something for when there's flooding. From the point of view of the turtle, it probably affords them the ability to just hang out after they hatch, ingest some more of the yolk that they still have attached to them, and hide out from predators until they get at least a little bigger. I mean, by the time they hatch, they're silver dollar size. They're still little protein packets for any passing raccoon. But they're probably, there's some reason that selection has favored them doing this, because not all turtles do this. Here's a female digging her nest. So as with all turtles, the nest is, the size and shape is dictated basically by how long and strong her rear legs are. So she'll find a good spot and then she'll start digging, and this can take 15 minutes, more like an hour. Sometimes she'll pee on the grass if it's too, or on the gravel or sand or railroad embankment or the side of the road to help loosen it up. And then here's a cute little picture of a little hatchling. Again, these are about an inch in diameter. And then that's my little hatchling holding a little hatchling up at Port Deposit. Thank you. Okay, so a little bit more about our particular population, by which I mean the Maryland northern map turtles. You can see that there are lots of records along the Susquehanna River basin. Here's the Susquehanna, it goes on up into New York. It drains a huge, something like 17,000 square miles. And this is the Juniata River, where they also inhabit. Let's zoom in a little bit on the Maryland portion of the Susquehanna, just a little bit of it, like 18 kilometers. You can see the state border there at the north. And then down at the southeastern corner of the map is the Chesapeake Bay. And they're sometimes spotted out there, like swimming out to, this is across from, what's it called? Elk Neck State Park. But as far as we know, they don't do any nesting out there. So this is what they have in terms of places to go in Maryland. Now, there's no hard border, of course. There's no check station there at the border where they can't go up into Pennsylvania. But from a management perspective, this is what we have. There is, however, a physical barrier right in the middle. You can see Highway 1, just like out here. Keep going. You will cross the Conowingo Dam. And that probably is a hard barrier to the turtles moving. Is radio telemetry data suggests that they do not cross this river, this barrier. Sometimes the radio telemetry data will show that female turtles will go inland quite a ways, sometimes up to 1 kilometer. You know, they're trying to get to the right height. So this is, they want to lay their nests so that their nests don't get flooded. So they've got a year. Those hatchlings have a year in there, and they don't really need to get flooded, or they probably die pretty quickly. But none of those data suggest that they actually climb up and go around the dam. This has strong management implications. For one thing, it suggests that there's no genetic connectivity above and below the dam. So perhaps here in Maryland, we actually have 2 populations. Yes, sir. Yep, we have radio telemetry data from both above and below. And this map doesn't really do it justice, but there are lots of dams on the Susquehanna all the way up, and there are turtles pretty much in between all of the major dams. So instead of having 1 population to manage, suddenly the state might have 2 populations to manage. Stay tuned on whether or not genetically they look like 1 or 2 populations. It also means that each of those populations is probably about half the size. Suddenly we've taken a population of some number and we've cut it in half. And it's also not always nice and peaceful like this. You can see you can walk quite a ways out into the former riverbed in this picture, but it sometimes looks like this. When there's an excess amount of rain, they got to let it through the dam. You can't hold all that rain back. This is a quite large watershed. From the turtles' point of view, what the river below the dam can go from looking like this to looking like this. And remember, I said that the basking was an important part of their natural history for both males and females, in particular for the females. This can happen, we can go from that to this in the course of an hour. And if there's a lot of this during the spring, which can happen any spring is up and down all the time. If there's a lot of this during the spring, then it could be driving down the amount of basking that the females can do. And if it's driving down the amount of basking the females can do, it might be driving down the number of eggs that she can lay, or even the number of visits, the number of times she'll actually come up and lay nests. And if that keeps going for years and years and years, you can see what's going to happen. There are going to be fewer eggs laid. The population may shrink because of this kind of thing. Here are a couple of pictures. This is about 45 minutes apart. The dam has been opened a little bit. So these, I don't know, 8 or 10 turtles, sorry about the quality of the picture, but you can see this is the same rock. About 8 or 10 turtles have had to jump off. This is kind of like the Yertle the Turtle book. And 3 females are hanging on right before they started jumping off there. A textbook example of organisms moving uphill in response to changes in the environment. Okay, well, I told you we were going to talk about some solutions during this talk. Here's one solution that we have come up with, and it's pretty low tech. Build them something that floats so that when the water goes up, they can still get a ride. They use these, they love them. This is nothing fancy. It's just some plywood and some floats. And then you have to put a buoy on it according to Army Corps of Engineers, Coast Guard, USGS also has a say in this permitting, and of course the state. So the building little platforms are not labor intensive. What's really difficult about this is getting all the permitting, and then getting the floats to stay put. Because you can imagine when the river is raging, like you saw in that other picture, these often get washed downstream. It's really hard to anchor a large floating thing with a lot of surface area to the bottom of the river. So we're continuing to work on trying to get more of these deployed in the quantity that we need to probably be positively affecting reproduction of the turtles. A further insult to their reproductive abilities is nest predation. Now, nest predation is normal for turtles. It's been happening longer than humans have been around. But what matters is how much. Even in pristine environments, we see a lot of nest predation, often on the first night. Some populations, you'll see something like 90% of turtle nests are predated in the first night. And it's usually this culprit, the raccoon. And raccoons, of course, are what we call subsidized, by which we mean that when it's not egg season, then we as a society are feeding them year round with things like dumpsters and growing gardens and things like that. So where they're humans, there are often more raccoons. And so that's a problem for things like northern map turtles. Above the dam, so the dam now is in the top corner here. Above the dam, the nesting is pretty dispersed. There aren't these big congregations, but below the dam, nesting tends to happen in just a few concentrated areas, as far as we know. So this is putting fewer baskets, or putting more eggs in fewer baskets. So the sites I'm going to discuss are at Octorero Creek, which is right here, and Wood Island, and at Port Deposit. This is the town of Port Deposit over here. This dot is some condos where they like to nest in people's front yards. And this site down here is one of the few other places along this entire side of the river where there is a beach where they could go up. When they were building the dam, they built railroads along both sides of the rivers, and that railroad still exists on this side, and it makes it hard for the turtles to get up and nest. So below the dam, that means that they're concentrated and probably have been for 100 years or so. We have some data on this. Down at Port Deposit, there are not a lot of nests, but what's weird about that site is that despite it being a town with a lot of dumpsters and people, we don't see any nest predation down there. Counterintuitive. That's not what the book says should be happening. Up at Octorero Creek, there are a lot more nests, and nest predation can vary widely from year to year. You can see from 30%. Some years it's a total loss, a total wash. No hatchlings emerge from the ground. Not even any hatchlings survive to become hatchlings. Not even any eggs survive to become hatchlings. And then at Wood Island, we have sort of a medium amount of nesting, but again, nest predation can be really high, north of 95%. Octorero, just based on the size, is probably the most important nesting site for the lower below dam population of northern map turtles, but it can have total nest failure. But even when there isn't total failure, modeling suggests that for turtles, most populations need to have something like 40% nest success or better in order for the population to persist. So if it falls below that, then over time, that might be 20 years, might be 100 years, the population is just not going to be able to sustain itself. It can't do what we call recruit new individuals into the population. There are several things that we humans can do. We've tried a couple of these and are in the process of trying a couple of these for our population at the Susquehanna. The first of these is habitat management, or habitat modification. This is at Octoraro Creek. The picture up at the top is what it usually looks like about April when I took this picture. And there's not a lot of beach that you can see in this picture. There are lots of invasive plants that are brought down by the flooding. The seeds get deposited in here, and there's a little bit of open sunlight there because it used to be a beach. And things grow up really quickly. And this makes it really difficult for the turtles to nest there. I mean, they're really strong, but they can't dig through 6 inches of tangled roots. So what we've been doing, what we've been doing experimentally, and we hope to sort of institutionalize, is this. So we get a landscaping company to come out here and cut down everything. And sure enough, not only does that encourage nesting, but it can decrease raccoon predation rates. I'll show you that in a minute. Another one of those things on that slide that I showed you with the options you have for dealing with nest failures to get rid of the predators. This is a problem because of the optics of what you do with this cute, smart, fuzzy mammal after you remove the raccoon. So if you remove raccoons, and we know this from lots of data, from lots of studies, if you remove the raccoons, it's not very long before some more move in. They make a lot of babies. They can move quickly. More raccoons will move in. So you kind of have to constantly be trapping these and taking them out. And then if you take them out, are you really going to take them out? Because the optics for that are really bad. You know, in a lot of places, in some areas, they are managed just by killing the raccoons. But most agencies, and certainly private corporations like Constellation that runs the hydroelectric power plant on the dam, doesn't want to see a bunch of raccoons being killed, because I can imagine how that might play on social media. So this is technically a management recommendation, but nobody is doing this on the Susquehanna right now. Another threat faced by northern map turtles on the Susquehanna is human recreation. I alluded to this a little bit on Wood Island, where one of the problems is the raccoon. Another problem on Wood Island is the presence of humans. Wood Island is part of Susquehanna State Park, and even though people are not supposed to go out there because of the danger of getting out there if they open up the dam, people do it anyway, and they do it all the time. Dr. Seal had some little thermometers soldered to a little computer chip in a bunch of the nests on Wood Island, and they just, when you go dig them up, and you can plug them into your computer, and it tells you what's been happening with the temperature, and you can see it going up and down a little bit every day. One of them went up and down, and then one of them spiked, and all we could figure is that someone was building a campfire right on top of that turtle nest. And they bring their dogs, and that they probably leave their trash, and that encourages the raccoons, et cetera, et cetera. So that's a little bit of human recreation that's not good for the turtles. And the other is just people being around. This is in the town of Port Deposit. This is that little stretch of beach, and I mean about 50 feet wide, where the turtles can actually get up on the shore and nest. And they do it right here in town. But they're not going to do it when there are people hanging out there on that nice sandy beach, as people tend to want to do. Note the marina that's in the background of that picture. Susquehanna from here south down to the Chesapeake is beautiful and navigable and popular. And so there are a lot of boats out. Here are some data we collected about 10 years ago. Mean number of boats per day that go by this particular spot, actually a little bit upriver of Port Deposit. And you can see, as you would expect, there are more boats in the warmer months. And then we also counted the number of disturbances, and that aligns pretty well with the number of boats. See, September and October are popular with the human boaters. They're also popular with the map turtles. And it's also uneven across the sexes. So in June, remember, the females really need to bath so that they can stage, they can get their eggs ready for laying. They are being disturbed at a higher rate than the males are. And they're particularly disturbed by slow-moving boats. The speedboats, there are only 2 in this particular study, but just from anecdotal evidence, a speedboat that goes by, the map turtles don't care. They'll stay up on whatever platform they are basking. But if something slow comes along, like somebody in a fishing boat with their trolling motor on, They don't like that. They will jump off. And if another slow craft, many times has people researching map turtles, comes by, they will also jump off into the water. They are disturbed. So human recreation is another disturbance for them. Another one is habitat loss or habitat alteration. Probably, as I mentioned before, one reason that the nesting is so concentrated here below the dam is because there's a lot of the river, especially on the Cecil County side, that they just can't climb up. And that's probably been the case since they built those railroads for the construction of the dam, in part for the construction of the dam 100 years ago. So it's likely too steep for them to get up, or there's things like this. This is Marina Park in the town of Port Deposit. This stuff is just too hard for them to get up. And here's our beach again. This is in town in Port Deposit. Note the marina back there again. This is what it looks like most of the year. There's a bunch of stuff that washes up here on this beach that comes down even through the dam and prevents what little beach they have down here from being accessible. This one's fairly low tech. Every year in May, a local nonprofit group called the High Five Initiative comes out and they help us clean up the beach. Often they bring a whole army of Girl Scouts, and they're great because they can pick up all the little pieces of trash that accumulate. Okay, so those are just some of the threats and some of the successes that we've had with the map turtle on in the Susquehanna. Here is some ongoing work. This is back at the nesting, some ongoing problems that we're addressing, or just starting to address, or will need to soon. One of them is the flooding. So this is back at Octorero Creek. This is the primary spot for turtles to nest below the dam. And you can see, this is a picture I took, I think, in March or so. You can see where the high water mark was from flooding that year. And that would have flooded out any nest that was below it. The river is this way in this picture. And if nests are flooded for, people have actually done this experiment where they flooded turtle egg nests. We have not done that here. And to my knowledge, it's not been done map turtles. But if you flood turtle eggs, they need to breathe. They need to get oxygen, even though they're buried in the ground. If you flood them for very long, the eggs just die. So it doesn't take very many flooding events to cause this. There's also the invasive plant problem that we have. 1 year after having a total nest failure, nothing emerged in the spring. We dug up some of the nests, and a lot of them had invasive plant roots growing through them. And we don't know if the roots were going after already dead eggs just to get nutrients, or just by coincidence they grew that way, or if the roots were actually killing the nests, because these are invasive plants. So we often don't know exactly what they're doing in this habitat. So this is something we're working on. So you can imagine what we're thinking. Trying to make some hills so they can go up higher or something like that. We've experimented with that a little bit down in the town of Port Deposit by basically bringing in dump truck loads of sand, dropping it there so that the turtles will go up a little bit higher and maybe get above the high flood stage. This is going to continue to be a problem. The town of Port Deposit is soon going to be trying to get a grant to help them deal with flooding in the town, because they have a couple streams that come down into the Susquehanna and comes right through town. Main Street has flooded several times in the last 10 years. Marina Park has been totally underwater several times. So it's not good for the turtles, but it's not good for the humans either. As I alluded to earlier, we're also starting to use some molecular techniques to learn about things that we can't from other means. For example, time and time again in science, now that we have all these DNA technologies, we have had to unlearn old assumptions we made about organisms in the wild doing things that we didn't know they did. The classic example of this is that ornithologists forever thought that almost all songbirds were strictly monogamous. And then we started genotyping all of them and doing all the daytime TV trick, trying to figure out who's related to who. And we see multiple paternity across all kinds of songbirds all the time. So just because you can't see it in your binoculars doesn't mean it's not happening. So when I said to you earlier that the turtles don't go across the dam, that may or may not be true. We may just not have put the radios on the right turtles. So they might actually do that. But the DNA can tell us about some of these things. It can tell us who's related to whom, like I talked about with the box turtles. It can tell us how many populations there are. It can tell us approximately how big the populations are. It's really hard to go out and find all the turtles in a river that big. Okay, what's the question? As a species? That is a good question. I don't know, but I can find out for you. I have a guess, but I want to check it before I check that, before I say that number. So I'll find out and let you know, and you can get back with that person. So we're doing some population genetics work. This is just a slide to show you about how lo-fi some of our methods are. How do you go about getting your hands on a turtle? Where we spend a lot of time out there doing this on warm summer mornings in June and May, trying to figure out the particular trees or rocks that the turtles are choosing to climb up on that day. And then when you think you have it all figured out and you know what those turtles are thinking that day, you go out and you set up these things. And it is pretty lo-fi. It's just a bunch of chicken wire. And you try to hang it under those trees where the turtles were coming up. So you get in your kayak and you drag this ridiculously large thing out there in your kayak and you tie it to this tree where the turtles were basking before you came over there with your kayak. And then you go back to a safe distance and you get on you watch from your scope. And then if you're lucky, you get back in your kayak when they come back up. And when you approach them, they dive off the water into that big basket, if you're lucky. This is a really inefficient method, but it's the best we've come across for a small turtle in a large river. There are lots of ways to trap turtles. You can put out hoop nets and things like that. It just doesn't work in this big river. So that's how we get our hands on samples for doing things like taking a genetic sample. Another project that's coming up, you may have seen this, there's been a bunch of funding released in the last couple years on infrastructure. One thing that needs to be replaced is the Amtrak bridge over the Susquehanna River. It is aging, starting to show its age. So there's now money to replace that bridge. But this is going to be a multi-year effort. And we're talking on the Cecil County side, in particular down near Perryville, where the bridge goes across, there are map turtles that we see out there. So in the future, we're going to have to do things like figure out where they bask. Well, we kind of know where they bask, but how we can mitigate that during construction, because they're going to have to build this sort of pier next to the old bridge where the new bridge is going to go. And this thing's going to be out there for years, and it's going to disturb the turtles from doing basking, which is really important for their natural history. We also don't know much about what they do in the winter. Up at Wood Island, we know that there are a lot of them that hang out there in the winter, but we don't know if those are the same turtles that bask down here in Perryville. And they can't just, because this is a state endangered turtle, you can't just do construction in the water. You know, they're going to have to build these dry docks where they're going to put the pylons for the new bridge. So they're basically going to make a hole in the water with some dams to keep the water out, and they're going to build the footings in there. And if turtles are used to spending the winter down there, then some poor scuba diver is going to have to go look for that. And then what do we do with the turtles if we do find them? We can't just put them in the fridge for a few years. So there are a lot of questions there that we're going to have to ask and come up with solutions to. Radiotelemetry is one way to figure out what the turtles are doing. This is, again, pretty lo-fi. You put a little radio on a turtle, and turtles are great to study, by the way, because you just glue this thing onto their nice ready-made surface. It's not that easy for almost anything else. You can imagine what it's like to study snakes. You can't just glue things to a snake very easily. But with the turtles, you just glue this radio transmitter on it, and it beeps once a second or so. The battery will last a couple years. And some poor student in the winter, not this nice, pleasant summer breeze kind of field work, some poor student in the winter is going to have to every day or twice a week go out and figure out where the beeps are loudest. And that's where the turtles are. So he or she will be on their boat circling around a lot in the winter on the Susquehanna River to try to figure out what the turtles are doing in the winter. They probably aren't just staying put in one spot. They're probably moving around, but we don't really know. Another project that's coming up, eventually we are going to have to dredge behind the Conowingo Dam and other dams. So 100 years ago, there was quite a bit of reservoir, by which we mean lots of room for silt that comes down from the basin off of crops, agricultural fields, mine tailings, urban areas. It's just been allowed to build up. And in some respects, the Conowingo Dam helps protect the water quality in the Chesapeake because it keeps a lot of that muck from coming out and coating oyster beds, and coating blue crabs. But it's building up, and there's only like something like 15% of the capacity left. So when we get these increasingly severe winter storms that we're going to in the next century, we're already seeing that in the town of Port Deposit, there's nothing for it. We're gonna have to start dredging that stuff out and doing something with it. We don't have a great handle on what's in there, how toxic it is. We don't know exactly what we can do with it, but we do know that map turtles can't be down there when they start dredging. Has anybody ever seen the kind of equipment they do for this high volume? Basically, they put a big wet dry vac down there, mount on a boat, and then they have a big tube that goes to the land or to a barge, and they just suck everything up. Yeah. It depends on what's in it. Maybe so. Maybe that we could solve two problems with one boat, I guess. Yeah. It depends on where you are, right? When it's flooded, that's going to depend. So above the dam, even when it's flooding, it's not going to fluctuate, not going to change a lot because it sort of spills over the top of the bathtub. So it's probably 30 feet, right behind the dam. But below the dam, you remember that picture when the water was all flooding through? It's really going to depend on which spot you measure. It could be 30 feet, and it's moving fast at that time. So the turtles are probably okay with hiding behind rocks or not getting totally washed downstream. At least the radio telemetry doesn't show them getting totally washed downstream. But yeah. Yeah. So this is something that's coming. Also, Constellation, which runs the hydroelectric plant on the dam, they don't own it, but they rent it, they lease it from the state to run that, are in charge of all the recreational areas upstream. Like if anybody goes and fishes up there, Broad Creek and Conowingo Creek, and they're getting silted up too. So you can't even get a boat to some of those boat ramps by about June. There's just this much water and this much silt. And we do know from the radio telemetry that like in Broad Creek, the turtles spend the winter in there. So you can't dredge in the winter. In other areas, they don't spend the winter. They spend the summer in there. So we're gonna have to do a lot more work figuring out what's happening there. And then of course, one thing we have almost no idea about is what is the health status of the individual map turtles in the Susquehanna. So this is a map showing the drainage for the Susquehanna River Basin. And this is just one of a whole series of maps I could show you. This one is for nitrogen yield in subbasins of the Susquehanna River Basin. So in some of these areas, you can see there's a really large load of nitrogen. And there's all kinds of stuff that comes washing down in this water. We have almost no idea if it's hurting the turtles or if not. We know from other systems that when you have, say, a lot of, you've heard about these forever chemicals, PFASs and things like that are found in plastics and things like that. We know that they're affecting the microbiome, say, of other turtles in ways that's probably making them less healthy. Like specifically like ways that they can store fat, for example. So using some molecular techniques, again, just going out and catching turtles with that ridiculous setup we have with the kayaks, take a little bit of blood, and you can do a lot of stuff with just like, a teaspoon of blood, trying to figure out exactly what's in there and what the pollution is doing to these turtles. Because of course, what we see out there are the healthy turtles, right? They're the ones that come up and bask. They're the ones that when we capture, they look pretty good to our sort of untrained, non-vet eyes. But we don't know what's going on inside the turtle. And we don't know about all those turtles that we never see because they're sick and they don't bask or they just die and get, eaten by scavengers at the bottom of the river. So we're probably seeing the healthiest of the healthiest in our samples. So there are problems with that, but this is a project we're just starting to work on writing some grants to try to figure this out. So maybe this summer we'll start getting some turtles for this project. So the northern map turtle on the Susquehanna is faced with all kinds of threats and stressors. I've talked just about just a few of them today, but if there's something that is threatening a species in this world, it's probably threatening the northern map turtle here on the Susquehanna. And we have all these different players. You know, I mentioned all the permits we had to get to put out those buoys. It's complicated. So sort of one bright spot we have is in the town of Port Deposit. That town, you know, most of the time, if a municipality learned that there was an endangered species found in their town, they would want to stay away from that because they don't want to impede their ability to develop, for example. But the town of Port Deposit has embraced the northern map turtle. They've actually made it their official logo. When you drive through downtown Port Deposit, you'll see the little flags hanging off of the lights, and they say, you know, shop, dine, and eat in Port Deposit. And there's a picture of the map turtle. This beautifully restored building that you can see back here. This used to be a gas house. So about 130 years ago, somebody built this, and they stored liquefied gas there to power all the lights in town. About 10 years ago, it was decaying. No one was doing it. It was owned by the marina. The marina gave it to the city, and the city found somebody to fund this beautiful restoration of this building. And they stripped it down to the rock walls, and they rebuilt it in the original architecture. It's got beautiful wood vaulted ceilings, etc. The first floor they used for the town museum, because tourism is an important part of the economy here in Port Deposit. And then they let us use the second floor as our research field station. So Alex has been up there, spent many hours up in there. We use the area for research, but we also use it for outreach. So we will have pretty much every summer, we have some Towson University students who get paid to go sit up in there on Saturdays and Sundays when the town is kind of bumping. And what you can't quite get an appreciation for, well, maybe you can from this picture. So this is, you walk up this promenade, and then right off the picture here is the gas house. And then if you keep walking up the sidewalk, you get to the bars and the restaurants and the marina and things like that. So there's a lot of foot traffic in this part. Well, the town has let us use this second floor for research, but also to do lots of extension and outreach. So we can touch a lot of lives, tell people about the northern map turtle, and in turn teach them about some of the ecology of the Susquehanna River. But I've sort of left this whole trail of names that are involved in protecting the northern map turtle in the Susquehanna River. There's, you know, Constellation there, who has some in as part of their relicensing of the dam that got relicensed a year or two ago. They have to protect things like the bald eagles and the eels and the shad, and the map turtle is on that list too. There's the town of Port Deposit. There are two different county governments on either side. There's the Maryland Department of Natural Resources that is in charge of permitting anything that might affect the state endangered species. Now we've got Amtrak involved because they want to do this construction. They want to rebuild that dam. So it's a complicated story, a lot of politics. But as you can see, we got those buoys out there despite all the permitting. So there are some bright sides to it. We've done some of the habitat management on that beach at Octoraro Creek. So we know that in addition, when you take the landscaper out there and you cut down all the invasive plants, it encourages nesting. It also decreases predation, raccoon predation, for reasons we don't quite understand yet. And of course, it's also an excellent place to train a lot of students. One of our missions at Towson University, or really at any good university, is to train students in real world problems, and they get quite an education. There are actually some living quarters in the back of that field station where students can spend the night. There's just some bunks and a full bath and a little dorm fridge and a hot plate, a microwave maybe. But there's Wi-Fi and air conditioning, and so students can hang out there and do research all summer, and those are real job skills that they can get. And then they also get exposed to what conservation is here in the 21st century, which is lots and lots of compromise with lots and lots of people. It's very social. And then of course they get to learn how to take blood from turtles, and they get to learn about involving the community. They get some skills like this they can put on their resume. I know how to do biological surveys. They know how to deal with chicken wire. Okay. This is the end of my talk. This is a cute little hatchling. This QR code will take you to the Towson University webpage that is about the field station. You can learn a little bit more about the field station and about the town there. So I'll take any questions. Yeah. The females get bigger, much bigger. Yes. The largest one I've ever seen, probably about this big, quite hefty. And they've got serious jaws. Remember, they can eat things. Not as big as snapping turtles, and a little nicer than snapping turtles. Not as dangerous for your fingers, but they will still hurt. Yes, sir. Which one? Lots of different things. So that nesting stuff was funded by Constellation. It's now called Constellation. It used to be called Exelon. Some things are funded by the State Highway Administration. The interns who spend the summer up there, they're funded by Towson University. I'm applying for a couple grants. I shouldn't say I have them yet, but some of them are funded by Animal Welfare Institute, which is a nonprofit. Another one I've written it for comes from the college. There's going to be funding that is federal monies for this Amtrak thing. That is federal money going to Amtrak to rebuild the bridge. What else? Yeah, it's kind of all over. So private or public companies, but also government organizations. Oh, I shouldn't forget the DNR funds some of this stuff. They're funding the project looking at the population genetics, because when we have the answers to that, we can tell them, hey, you have one large population here in Maryland, or we can tell them, oh, you have two, each one's smaller. They need more aggressive management, maybe. So a bunch of different funders. Yeah, yes, sir. Mm-hmm. Yep. Yep. Certainly, because all those things we can expect basically any perturbation to affect the microbiome. That's why the microbiome has the potential to be a really important management tool. Also has the potential to be really useful for us humans. But we also know it changes a lot. So if you get a new roommate, your microbiome is going to change. If you get a dog, it's going to change. If you're a vegetarian, it's different than if you're a carnivore, even in the same household with the same dog. It changes by where in the world you live. Changes as you get older. It's different back here than it is here. So it's really hard to figure out what healthy is. Exactly. Yeah. So we might find the microbiome is different above the dam and below the dam. We might find that it's different in males and females, especially given that they have different diets. Yes, it's all possible, but we don't know any of that until we do this basic research on the first thing is to just sequence a bunch of swabs and figure out what's in there. Okay. Okay. The turtles are marked by we drill into their marginal scutes. So again, turtles are relatively convenient, I guess, in that you can give them individual marks. So you can see these each of these little blocks here is called a scute, and those appear in the same manner on pretty much every turtle. And you can mentally assign a number to each one of those scutes, and you can give it basically a barcode. So on the first turtle you pick up, you drill a hole in this scute, and that'll last for decades maybe. And then the second turtle you pick up, you drill a hole here and a hole here, and that's turtle number 2. And the third turtle you pick up, you mark, well, this one and this one. And they have so many different scoots, and they have 2 different sides, and you can use the scoots on the back. So you could, in theory, come up with thousands of unique barcode IDs for them. Are they protected in other states? I don't think so. If you look at the IUCN Red List, they are least concerned. In many of the states, like Indiana, where they exist across the state, they're not listed at all. So if they're listed anywhere else, it would be like here on this edge of the species range, like perhaps New Jersey, because there's just that little population. Yeah. Okay. Okay, good. Yes, sir. Sure. Yeah. There are several places that are raising hellbenders in boatloads. When I was at Purdue in Indiana, I was raising maybe a canoe load. We had just a few. There's only one relict population hanging on in Indiana, kind of like here. And they're all old individuals and they're skinny, but they're Eastern hellbenders. They don't seem to have this lesion problem. So neither did we see it in captivity. The place with the largest captive Ozark hellbender is the population is the St. Louis Zoo. They're actually good enough. They've built this little fake stream in one of the yard, and they can get them to actually breed in captivity. That's different than going out and finding eggs, bringing them back in, and raising them, which is what we did at Purdue. They don't seem to be having that problem in the captivity. Yeah. 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 And that might be a lot of birds are doing that too. They carefully pick them out. Yeah. Right. That I think so. I never saw that site, but I'm told it's about 2 kilometers upstream. This turtle came from there. So the food available to the turtles in Deer Creek is different than that where most of these samples came from out here in the main branch of the Susquehanna. In addition, in particular, there seemed to be a lot of this invasive Asian clam for whatever reason in there. No, I didn't get to sift through any poop. Again, this is Dr. Siegel. Yes. That's right. And this is something that in the future we might do this experiment again, but we might do it with a DNA-based thing. So instead of picking through the poop and because what kind of things are going to survive going all the way through the turtle and show up in the poop? Is it the soft, squishy bodied things? No, it's going to be the things that have shells like arthropods and the clams and the snails. But if we took all the DNA out of that poop and sequenced it, you can get a less biased idea about what they're eating. So that might be one of the other projects. If you want to take on that project, then you're welcome to do so. No, you don't want to deal with the poop. Yeah, I often get pooped on when I handle a turtle, but the map turtles, yeah, pretty much everything. I didn't show you this data, but the more specific breakdown of the stuff we find under the scope is 99% animal matter, a little bit of plants, and that might just be accidental. And it starts to what? A toad? Oh yeah, toads. If you picked up toads before? Yeah. They pee on me still to this day. They're hard to do. But yes, so in particular, Dr. Siegel did a population estimate of females, because you can grab them and mark them in greater numbers because they come up to nest. The females below the dam, he got an estimate of something like 60, plus or minus 20 or 30. So there's a big range of error for those. One thing we might do down at Perryville with the bridge project is to spend a lot more time out there with spotting scopes, just counting turtles. You go out for a set amount of time and look at a set number of trees, deadwood, rocks, or something like that, and start to keep data. And with those kinds of data, you can get decent statistics. And that's another thing that we don't quite have the numbers to say anything about it yet. So we need to redo that survey again and see what the numbers say. I think they're like the hoop traps. They're just really inefficient. I tried. Right. And then right. I tried it a little bit this last summer and they totally ignored it. But it was in an area where we saw them basking anyway. So they had more trustworthy things to climb. You know, like the snags they were used to. Yeah. Right. So we could just do this. Yeah, that's something to try. It's such a hassle to get the permitting for those. But that doesn't mean we couldn't do it. We could put a little trap at the top and they climb up there. Right. Well, they maintain it, and so they keep it clean, and it's big rock, kind of like this. They do, especially right behind the field station. They nest in that stuff sometimes, but it's often too steep for them to climb up. And then they encounter this rail, which they can probably climb over, but maybe not. It's just one of those. It's not the nice sandy, smooth, low-grade beach like they have at Octoraro Creek, at Wood Island, and in the town of Port Deposit. Yes, sir. Let's no. They as far as we know, no. Dr. Siegel did a survey to look at that, and he sees them using the water around it, but not nesting there. Again, as far as we know. So they could be, but because that's part of a wildlife national refuge system, right? I think they wanted to know at one point, and it did not seem to be a big spot for nesting. But I've not been on that island. Is it what's the bank look like? Are there nice low sloped sides to it, or is it okay? That's another thing. Okay. Mm-hmm. On the island or right off? Okay. Yeah, it's probably worth another look. How much open sand is there? Like on Wood Island, they like to go to the spots where there's some sun. They don't want to. They don't want to nest in the areas that are too shady because they don't. Okay. Yeah. Yeah. Well, might be worth another look, especially if we're going to be down that way, looking at the railroad bridge. Not yet, not with this species. One thing we do sometimes, like when we're trying to figure out what natural predation rates are, is when we at Octoraro Creek, sometimes we put up a hunting blind and we hide in there and we watch the turtles nest, and then we know where they nest and we can put a little cage around the nest to keep out the raccoons. So that's like the easiest thing to do. Yeah. Yep. Yeah. Yeah, and that's certainly a relatively easy thing to do. If we know where they are, then we could move them uphill is another thing we might end up doing. We're hoping to soon start surveying Octoraro Creek again more intensively to see if, like the last time, I think in 2016, we looked at it, there was total nest failure. If that's normal, then we're going to have to start doing things like this, or even taking them to zoos or something. If that was an anomaly, then maybe we don't need to do that yet. Sure. At Octoraro Creek, the site is actually facing the Susquehanna, not up that creek. But in other creeks, we see them, like Broad Creek, we see them going in, and Deer Creek too, a couple kilometers. So they're not just in the main trunk. We always say that their range is the Susquehanna and their tributaries. Yes, sir. Good question. I don't think anybody knows a whole lot if they're actively feeding down there. I mean, it would make sense that they might eat a gnat or a worm or something that presented itself in the burrow. But at least the book will say that they subsist mainly on their yolk while they're down there. And they're pretty metabolically chilled out because much of that time is winter. So they're probably not doing much and they don't need much to eat. And they're, when they do come out, they're still silver dollar size and they're squishy. They haven't done a lot of growing. And then that first year, they put on remarkable growth, especially the males. In that first year, though, by the end of the summer, they're probably 4 times the size. But when they emerge, they're still look like lots of snapping turtle babies and things like that. But all we need to do is put some cameras down there and watch them. We could probably learn all kinds of stuff about that and about the tree roots, the plant roots growing through it. There's a lot of work to be done. Yes, sir. Yep. Yeah. Yep. There's a recent really cool paper with some really cool footage from Canada where northern map turtles live in areas that freeze over every winter. And what's interesting about that is that they do move in the winter. If you look at the footage, you're like, oh, that looks like a normal turtle movement. And then you see the footage is slowed way down, sped up 10 times or something. So they're really slow. And the movements that they're doing are they're trying to get from the 4-degree water down to the 1-degree water. Because if they stay at 4 degrees, they're too active metabolically to survive the winter. They'll drown. But at 1 degree, they can be so chill that they can survive until the ice up there melts. They're doing a little respiration in their mouth, probably a little in their cloaca, but mostly their strategy is to be really chilled out. They're probably doing something different here because the Susquehanna doesn't freeze over all winter. But like I said, that's one of the things we need to learn more about. We need to subject more students to doing radio telemetry in the winter to find out what they're doing. Or if I have my way, we'll get some big grant. Recently, drones have taken up a lot of this radio telemetry work. So you can send the drone out and they're much more efficient at tracking down a signal. But can you imagine what it's going to look like when a drone is flying over the Susquehanna River? How are people going to respond to that? So that's another challenge we have to think about. Yeah. Mm-hmm. There's probably, he was asking about if the camera data show the raccoons not eating as many eggs because they are exposing themselves, they're going out into a more open area. I think there's probably some of that, but it's not it's like 20 feet wide and maybe 200 feet long, the area that we clear. So it's not like it's an open sky, but something is causing a little bit of reduction in predation when we do clear that. And the rest of that area is all nice, snug forest where they can hide. So maybe they're just preferring to stay a little bit more hidden. The camera, we have put out game cameras for a lot of that. It's hard to tell from that kind of information what the raccoons are thinking. I'd have to think about how we would do that. Yes, sir. You can contact me, or you can go to that QR code that's in the last slide. Sorry, I subjected you to so many slides. And there's basically the same contact information. There's certainly some, depending on what's going to happen this particular spring. That depends on some permitting and projects and grants. So it could be that we could use people to go and look for turtles, like on the nesting beach. We often have, when things get really hopping in the middle of June, we often get a lot of phone calls. Like somebody who owns a condo in Port Deposit or Perryville will call us and say, "Hey, there's a turtle nesting in my yard. You want me to do anything about it?" And we take them, a little cage to put on there to keep out the raccoons, because in some of the condos they do have raccoons as opposed to there at the field station. And then if anybody else, if anybody has other special skills that maybe could help the project, then we're certainly willing to entertain those. Unfortunately, we don't have a great right now. We don't have a program that's kind of like the sea turtle volunteer programs that require hundreds of people to walk the beaches at night and mark where the nests are. For one thing, these guys nest during the day, and for another thing, the getting permission to go to those particular sites is not as straightforward, unfortunately, for some of them. But especially if you live up in the area, there are send me an email about 2 months and I'll have a much better idea about what we're doing this summer, because I'm still trying to get the grants together, and that determines what we're going to do this summer, because we've got a lot of fingers in a lot of pies. Other questions? Yes, sir. Can the eggs? We don't know. We don't have a great answer for that for northern map turtles. In other turtle species, it can be like half an hour or maybe a few hours that they can be, underwater or soaked, in a beach. This is sandy soil. So even if it rains really hard, the soil should continue to drain. So the water table should be below where the nests are. So even in a hard rain, they shouldn't totally flood, assuming the female nested far enough up the beach. And they often are not happy with us here at the field station. We have this fence here, and it's partly to keep the humans out of the nesting area, but it's also to keep the turtles in the nesting area. Because if they go past the field station, their choices are a parking lot, a railroad embankment that's right behind there, and roads. And there's always a UPS van or something, or people driving their boats around or something. So we try to keep them in that little yard, but they don't like it. You can see them constantly probing the edge of the fence, trying to get to higher ground, probably. No, not since I've been working on this project, and that's 3 or 4 years. There probably is some, I'm sure, but I personally haven't seen any. Have you seen any, Alex? Yeah. So they have one of their adaptations to living next to a river is to try to nest as high as possible so their eggs don't get flooded. Just saying. We leave it up. Right. We tend to leave them up because if we take them off, it's easy to lose track of where they were. And in the spring, depending on what we're after in terms of data, in the next spring, we might want to keep them on there because then when the hatchlings come out, we don't want them to escape before we count them and see how many survived. Sometimes we dig up the nest to see how many didn't survive. So we tend to leave them on there, especially if the area is protected. Like this is behind a fence and it's in town and there's security cameras up there. So we leave the little cages on there. The caveat is that you don't want to cook the little hatchlings when they come out too. So we have to be on our game in the spring and watching those because one day all the hatchlings will come out. We want to collect our data and then let them go. I don't know about hatching. We tend to see them, and I don't know about the emerging either, but we tend to see them when we're out there before noon, before 1 o'clock, I would say. But I've seen the hatchlings all time of day. Sometimes they seem to wander around. They don't seem to quite know in which direction the water is. Often people will come up to us and say, I saw a hatchling headed for the road. And they turned it around. So, I don't know if that's been up for an hour or a day. It's kind of hard to tell. I've never actually seen them crawl out of the nest because I imagine that's pretty quick. I would guess that here the average clutch size is probably 8 or 10 or 13 or something like that. It's one of those things that probably varies a lot over their range. But Brian would have some of those data since he actually did some of the digging up of the nests to figure out exactly how many were laid. Yes, sir. Yes and yes. We're using the microsatellites just because this was supposed to be a grad student project, and I didn't end up getting a grad student this year. So a really talented undergraduate student took the project. It was just easier for us to use the microsatellites because they've been used in this species before, and they're really easy to do. They're really easy to work with in the lab. If you've ever worked with microsatellites, they sometimes work, sometimes don't. So we have 5 that have been used in map turtle work before, so we can compare allelic richness and heterozygosity and things like that. We thought long and hard about doing, about doing the RADseq stuff because you can get so much more data from it. But given our time frame, we went with the microsatellites. But we're sequencing it on a myseq, so we're going to get way more data than we need. But that's fine. Yeah. Right. So in the future, we will probably switch to the RADseq. One thing that's nice about the RADseq sequencing that we're talking about, in addition to being able to get data on questions like how many populations are they and who's daddy and who's related to whom, you can also learn a lot about certain genes that are under selection. So I can imagine in a population like this, when you do those kinds of studies in turtles, genes such as hypoxia tolerance genes are often under really strong selection, meaning that any small mutation in those kinds of genes might lead to a dead turtle. So when you do RADseq, you get a lot more information, but it's a little more technically difficult to do that kind of DNA sequencing. So for this project, the microsats are what we're doing. You're welcome. Thanks for having me.